Hey everyone. So I'm talking to you today from Wurundjeri land. I'd like to pay my respects to elders past, present, and emerging. Okay, brilliant. So I'd like to start off this talk asking this question about what is dispersal. And it's a question that if you ask different types of biologists, you can get quite a different response. So in fact, when I posed this question to my own research group, I got about half the answers were something along the left-hand side and half of them were along the right-hand side. And the idea is both of these responses are correct. You can either see dispersal as something an individual does as it moves around during its life, or you can see dispersal as something which occurs above the level of the individual, which is a sort of an evolutionary process that is so wherein you could see, say, the spread of a new invasive species is similar to the spread of an allele moving through a population. And now both of these are correct. They've both been definitions that people have used for a long time. And the reason that they're both around really comes down to the fact that you investigate them with different types of data. So if you have, if you're going out and looking at individuals and observing them, then you're going to have an individual-based approach. If you're tracking, you're inferring what's going on at the individual level through observing overall population changes, then you might have something that's intergenerational. Now, in this talk, I would like to put it to you that if you use close kin dyad data, as the sort that Veronique's just gone and described, if you use close kin dyad data, you can investigate dispersal from both of these directions, and accordingly you can get dispersal inferences that are interpretable under either of these frameworks. So close kin dyads are just any pair of individuals that are close relatives. So basically, because they're close relatives, it means if you go back in time, you go back a generation, 2 generations, whatever, they're in the same place, and now they're in different places. So if you look down here, this is a top-down view of some apartment buildings in Malaysia, and this is looking at Aedes aegypti, mosquito, the dengue mosquito, and basically, these dark blue lines are connecting two ovitraps, so egg traps where we've collected eggs, collecting 2 traps where we found full siblings. So there are eggs in these different traps, and they're full siblings with each other. And the reason that they are in different spots is because the female mosquito has showed up later egg in one of them, flown to the next one, and later egg in the other one. So it's a specific individual dispersal we're inferring. But as I'll show later on, we can also use other close kin dyad categories to then infer intergenerational processes. So really today, this is why we're doing this whole research. This is kind of an empirical demonstration of the stuff that Moshe Jasper presented yesterday, which was on kin disperse, this framework of investigating dispersal. But this is part of an applied project, really, with in a collaboration with Cyrus. This is Dr. Samia El-Feki here. You can see in the photos holding an Ovi bucket in the left. So this is a collaboration with Cyrus. We're working in Saudi Arabia, in Jeddah, and we have a Wolbachia program we're initiating there to try and drive Wolbachia into the population of Aedes aegypti mosquitoes there, because if they have Wolbachia, they won't be able to spread dengue. So this is ongoing at the moment. Now, there's been a lot of talk about Wolbachia at this conference. I'm not going to go into all that sorts of details, but I'll tell you something operational about it, right? When you're starting a Wolbachia project and you're going into some big city like Jeddah, Jeddah's big, it's a really large place. You can't just, you're rearing mosquitoes in a lab. You can't just take them out and release them randomly across the whole area. Nothing will happen. What you have to do is you have to choose one area, and I've highlighted where we've got this area, Al Safar, I've highlighted in blue. It's about a square kilometer. You choose one area and you release loads of mosquitoes there and drive the mosquitoes into that population. And as a result of that, you can then get Wolbachia spread there, and then you can move on to another place. So what I was doing over there, where all these collections I was showing you pictures of, is going into Al Safar, using genomics and specifically using close kin dyad data to assess what can we learn about the Aedes aegypti population in this area. And specifically, how might this information, what information can we get that might help interpret the results of our first Wolbachia releases? How can we tailor this so that when we release elsewhere, we know what to do? And these are the sort of thing we want to look at are barrier effects. So you want to keep the Wolbachia in one place when you start releasing it. So it's useful to release in an area that's bordered by barriers. Al Safar region is bordered by roads. Some of them they're about 60 meters. Some of the smaller ones are only about 30 meters. And then you've also got dispersal distance. So the further mosquitoes are able to move in a generation, the larger the area that you want to release over should be. And then finally, population density. If you're in an area with high mosquito density, you have to release more mosquitoes. Now, using close skin diet data and the rest of the genetic data we had, we're able to actually get inferences on all three of these things. So first of all, we wanted to assess barriers. And now these are using close skin diets, and this is using an individual-based framework. So looking at the specific inferences of specific individual movements. So we've got the full SIB dyads, they have the same mother. When they're in different traps, they're collected at the same time. They are there because their mother has moved from one trap to another. This image here on the left, that is the southwest area of the Al-Safar site, and every square shows where we've sequenced DNA from a mosquito. Every red square shows where we found a full SIB that is one part of another. So there's another full SIB in another trap. So there's quite a lot of full SIB data we have. Now the idea is, if roads are no barrier, we expect to see similar distributions of full siblings, whether they're across roads, separated by a road, or whether they're not separated by a road. But if a road is restricting dispersal, we expect to see fewer full SIB dyads than expected when we're looking across a road. And that's exactly what we found here. So if we have these okay, so we have these colored areas here. This is basically the distribution of traps across different distances, and then we have every black circle indicates a full SIB dyad. So on the same side of the road, we've got these trap distributions and number of full SIBs we found, and we can get this. These are the observed values on the same side of the road, and then using these trap distributions, we can build expected values. We expect to see full SIBs in these sorts of distributions across this skinny western road, and then we expect to see this many full SIBs across this thicker, heavier southern road. And in fact, we see this slight reduction of the actual observed values across the western road and a complete reduction across the southern road. We expect to see 8 full SIB dyads. We found none. So we have evidence we can use this to detect barriers, but also we can get a sense of how strong the barrier is. It's not just an absolute barrier, it's operating to some strength. And the second thing here is going to be using, so this is now, we've got close kin again, but using it at the intergenerational level. So this is now, we've got full SIBs, we also have cousins, we also have other things. Now this follows from what Mosh was talking about the other day. And this is, so basically, this is a kin dispersed framework. I'm going to give a very brief introduction to it. Sigma, this is a variant, this thing here, this is Sigma. Sigma is like the holy grail for population genetic estimates, anything in spatial population genetics. This is Sewell Wright coming up with stuff in the 1930s, 1940s, and this is really describing a fundamental, the rate at which things change spatially from one generation to the next. It's a very useful parameter to estimate and describes the distribution of so dispersal basically from one life stage, say an egg, to the same life stage one generation later. So that's parent-offspring dispersal. And what Mosh showed is because these are variances, variances have something called an additive property of variance, which is where if you can get one distribution or a bunch of distributions and combine them, the variances just add together. Or if you have one big distribution, you can split it apart and the variances subtract out. So you can take this one big parent-offspring, the holy grail one, and you can say, well, let's break this down into little subcategories of dispersal. So we can have dispersal as a juvenile, dispersal while breeding, and dispersal while ovipositing. Now, this ovipositional dispersal, that describes the distribution of the full SIB dyads, right? So we've got this distribution of full siblings from before, let's say, this is a Malaysian one, and we have this is the sigma of the full SIBs, this is the ovipositional sigma. Now, what Mosh showed is in Aedes aegypti mosquitoes, if you work out the same thing for first cousins, the overall distribution, the kernel for first cousins, is basically exactly the same. It's one draw from the full SIB kernel and 2 draws from the parent offspring kernel. This is what we want to estimate. Remember, the holy grail, the sigma. We can just do this by working these two things out and subtracting this from this. It's quite nice. And when we do that, in Safar, we have our distributions here. We end up with an estimate of sigma of around about 150 meters per generation. What's nice then? So I said you can put this stuff, you can take these results and stick them into population genetic models. Well, here's one, neighborhood size, right? 1946. Neighborhood size is 4 times pi times sigma squared times population density. It's really talking about the size of the breeding neighborhood of the population. Now we can estimate neighborhood size from patterns of isolation by distance. And from that, once you know one of these, you can estimate the other. It's usually very difficult to estimate density. In this case, we can get a sense of density around about 1,500 mosquitoes per square kilometer. Maybe it's difficult to interpret exactly what that means, but we have data from other places now and we can compare them. So let's say here we've got Kuala Lumpur. So we're comparing open areas in Jeddah with a high density living in Kuala Lumpur. Dispersal is much higher in Jeddah, but densities are lower, which fits pretty nicely with what we might expect. So from all this, what we can get are these inferences. All right, so, well, what do we expect? Well, we expect that we've got a strong barrier down the bottom and we have a weak barrier to the west. And then so Wolbachia might spread out to the west, but it may also, we might see a bit more fluctuation of frequencies around these weak barriers. Dispersal is quite high, so we want to release across large areas, and that's nice because we've chosen a large area. You wouldn't want to release over a much smaller area than this. And then finally, we have low densities. The low densities mean you don't need to release as many mosquitoes, but you have greater stochasticity. So you need to carefully monitor stuff that it doesn't get reinvaded. Thank you.
Right, wonderful. Oh, yeah, stop chat. Yeah, cool. Thanks, Tom. All right, awesome stuff. Does anybody have questions for Tom? Anything in the chat? Whoops. Nope, nothing in the chat. Charlie, I see you're coming into focus.
Oh, that's just great stuff, Tom. But I was wondering how you relate that to multiple generations, in the sense that there could be storms or something that really sweep things across, and how do you deal with that sort of variability?
I mean, it's a good question. It's very difficult to really reconcile these two things. I originally started out thinking, oh well, I can break down this conceptual duality, but really there's no way of doing it, because sigma, I mean, you can't. For instance, I mean, in this case, what we've got is something like a stratified dispersal system is what you're talking about there, really, where you've got little moving around and then all of a sudden something moves from several kilometers away or hundreds of kilometers away and sweeps through. Yeah, I don't know, it's beyond me. But I mean, they're both worth investigating, and you can investigate both with kinship, but it's difficult to do. I mean, it's important to note here, really what we're looking at here is when I'm saying, oh, it's 150 meters per generation, I'm going to say that was for that specific generation. It's not, this does not say that this is in any way relevant to what happened before, but it is saying this is what's happening right now. So it's useful in that specific sense.
Yeah, really, really great.
Anything else? Oh, Ari, perhaps comment as to how people could use it in different insect species. That was actually going to be my question. So obviously this framework has been developed for mosquitoes, which have a very, distinct dispersal system, as you've outlined. So how can you apply this to other organisms? Yeah, well, I mean, for the individual based stuff, it's really quite simple, because that's you're really just being able to infer. You have to understand things like the mating system. So, for instance, in Aedes mosquitoes, you can say that half sibs are paternal half sibs, but that's generally quite simple. I mean, for working out how to break down the generational categories in the kin-disperse way, I mean, Moshe has investigated some of that, and I think yesterday he presented some stuff that I hadn't actually seen on butterflies, and he's also developed that for Antechinus, and so there are ways of doing it, but definitely require some specific thought. The idea, though, is this is really, the intergenerational stuff is particularly useful when what you're looking at is something that doesn't disperse that far, that you can set a lot of traps out within a certain area, but where you can't, it's difficult to directly observe what's going on. But so something that is moving around maybe cryptically, but is not moving that far. Yeah, cool. Thanks, Tom. Alex Piper has a question, or maybe we can say that to the
temporal comparisons of disperse. Have you done any temporal comparisons of dispersal distance at the same site? Not yet, but we're going to. Yeah, that's all I can say on that.
Oh, wink, wink, nudge, nudge. All right, so thank you, Alex, for that very snappy question, because that's perfectly tied us over to 4:30.